Hello everyone, this is Dr. Karen's again, and today we are going to talk about Chapter 6 in our textbook for microbiology for non-science majors at Eastfield. So this chapter talks about how different microbes grow, the types of nutrients and other physical and chemical properties that they need in order to grow and grow well. And then we'll talk a little bit about how to grow cultures of bacteria in the lab as well. And so this chapter is important kind of in two different ways. So one, it's obviously we're learning about the cells themselves and what types of things bacterial cells need in order to stay alive. But then also a more practical purpose is knowing those things allows us to maintain different lines of bacterial cells successfully in the lab. And so for us, obviously we want to be able to know how to do this so we can do our experiments. But if you're thinking beyond that, in a more professional microbiology lab, they need long-term maintenance of these cells or these cultures. So it's important to know how to optimize those conditions, how to optimize those nutrients so you can be successful. All right, when we're talking about growth, there's two ways to think of this. We could be talking about growth of an individual cell. We talked a little bit about that when we talked about metabolism. But then really what we're talking about in the lab is an increase in the population of the individual microbial cells. So when you're in the lab and you have a sterile broth with nutrients in it meant to feed bacterial cells, and you inoculate that broth with bacterial cells or place cells into that sterile broth, at first the only thing that's in that tube is going to be the cells that you just put in there, which aren't going to be very many. We want those cells to be able to live in that broth, use the nutrients in that broth, metabolize that broth, and then divide over and over and over again so we get a much more hardy population of cells and we can do more things with those. Some terms to think about when we're thinking about growth in this manner. A colony. A colony is a mass of cells arising from a single parent cell. So if you were to put a single bacterial cell on an agar plate, that agar containing nutrients for that bacterial cell, that single cell would use those nutrients, metabolize, grow, divide, and those new cells will do the same thing and keep dividing and dividing and dividing. And there's not a whole lot of movement of cells on top of an agar plate. So they're just going to keep piling on top of each other until you see, in a day or two, a visible mound of cells. And because they all came from a single parent cell, they are all genetically identical to one another. So being able to grow single colonies of cells is a really useful tool in the lab as a way to maintain what we call a pure culture of bacterial cells. If you're trying to study E. coli, you only want E. coli growing on your plate. You need to be able to grow single colonies so you can make sure that that happens. Another term, we've seen this before, a biofilm. So this is also a mass of cells, but they're not necessarily the same type of cells. And we don't necessarily purposefully try to grow biofilms. These happen naturally in the environment. And it really is a strength in numbers type of deal for these cells. We'll talk more about what that is in a minute. All right, growth requirements. So these is a summary of all the different types of nutrients that bacterial cells are going to need to do their metabolic work so they can grow and they can divide. So the first type of requirement is chemical and energy requirements, more particularly carbon, and energy. So we can classify organisms or classify bacterial cells based on what they use as their carbon source and what they use as their energy source. And so this chart here, this is figure 6.1 in the textbook. This is a nice summary of how we can classify bacterial cells and other organisms based on those two requirements. So on the left are the two different prefixes that we would use to identify the carbon source for that particular organism. And up on top, it's the energy source. So let's look at this. So photoautotrophs. The photo is referring to where that particular organism is getting their energy from. Photo means light. So think of plants. They can do photosynthesis. They're getting their original source of their energy is coming directly from the sun, and they're able to convert it into a chemical form that they can use. So photo means anything that has that term photo in it means they're getting their energy directly from light, from sunlight. Autotroph. Auto has to do with where their carbon source is coming from. And auto means carbon dioxide, an inorganic source of carbon. So a photoautotroph is an organism that gets its energy from light and its carbon from carbon dioxide. A photoheterotroph gets its energy from light, but hetero means the carbon source is from an organic compound like sugar, glucose. We just talked about that in chapter 5. That would be a carbon source. So photoheterotrophs, energy from light, carbon from an organic compound. Chemoautotrophs, so the chemo has to do with the energy source. The energy now, instead of light, is coming from a chemical compound. Autotroph means carbon's coming from carbon dioxide. And then what you might be most familiar with are the chemoheterotrophs. We are chemoheterotrophs. A lot of bacteria are also chemoheterotrophs. They get their energy not from the light, not from sun, but from a chemical compound like sugar, glucose, maybe lactose, some type of sugar. Their carbon source is also an organic compound, and it could be the same thing. It could be glucose, again, is serving as both an energy and a carbon source. So these are the types of categories that we can put bacterial cells into and other organisms based on what they use for their energy and what they use for their carbon. But there are other requirements as well, namely oxygen. And we can classify bacterial cells based on their oxygen requirements too. So in terms of how do we name these organisms based on their oxygen need? Those that need oxygen for life, we fall under this category. Many bacteria fall under this category. We need it for life. We cannot live without it. They, we, them, they're called obligate aerobes. Other organisms can die actually in the presence of oxygen, and they are called obligate anaerobes. And I want to clarify something here. It does not, for an obligate anaerobe, it does not necessarily mean that that molecular oxygen that's in the air that we are breathing right now, it doesn't mean that that form of oxygen is deadly to those organisms. The issue is molecular oxygen that's in the air can sometimes be degraded into toxic forms of oxygen, and it's the toxic forms that obligate anaerobes cannot manage. They don't have the right enzymes to get rid of or detoxify themselves from those forms of oxygen. We do have the right kind of enzymes to get rid of the toxic forms of oxygen, and so we can survive in molecular oxygen, and we need it too. But obligate anaerobes, it's not necessarily that molecular oxygen that's the problem. It's the byproducts of other chemical reactions that create these toxic forms of oxygen. What are those toxic forms of oxygen? peroxide anion, hydroxyl radical, singlet oxygen, superoxide radicals. These are forms of oxygen that usually have some unpaired electrons, and that makes them very reactive, and so reactive that they will oxidize things that we don't want oxidized, and it can cause damage to cells. So that is the issue with obligate anaerobes. There are some groups of bacterial cells that can fall somewhere in between those two extremes. I'm going to show you this image because I think it best illustrates the difference among them. So these are bacteria that are being grown in a special type of medium called thioglycolate. And in a nutshell, the oxygen that is in the broth, there's like a gradient of oxygen. There's more oxygen at the top of the tube than there is at the bottom. So obligate aerobes, since they require oxygen to grow, you will only see them growing at the top of the tube. And what that really looks like in real life is that you will see a cloudiness near the top of the broth, and the rest of the broth is clear. You can see through it. Obligate anaerobes are the opposite. They cannot survive in oxygen because of those toxic forms. So they want to hang out at the bottom of the tube where there isn't much oxygen there at all. And then, so those are those two extremes, but here are a couple of groups that fall in between those extremes. So facultative anaerobes, they grow better in the presence of oxygen than they do without, but they can tolerate environments without any oxygen in them. And so you see a grouping of these organisms near the top of the tube because that's the better environment for them, but you can also see growth throughout because they can tolerate environments without oxygen. And then aerotolerant anaerobes are indifferent to oxygen. It doesn't matter in terms of growth whether oxygen is around or not, so they will grow evenly throughout the tube. If you take this class in a face-to-face setting at Eastfield, you will do this actual experiment and you'll be able to see this growth pattern in real life. Other requirements: nitrogen. Nitrogen, most cells, if not all cells, are going to need nitrogen in order to make amino acids so they can make proteins. And also we need nitrogen for our nucleotides so we can make DNA and RNA. So it's a essential growth requirement. We all need it. We can't always use the nitrogen in the form that it's available, and so we rely, and other bacteria will rely on a particular group of bacteria that are able to do what's called nitrogen fixation. You probably learned about that in Bio 1. If you took Bio 2, maybe that's where you learn a little bit more about that. I'm not going to go into the detail, but it's basically a way that the bacteria can take the nitrogen in the environment and turn it into forms that our cells can use. And then let's see, some physical requirements. So temperature is one of those things. Think back when we talked about enzymes, and I mentioned that temperature can affect how well an enzyme can work because high temperatures will denature the enzyme. Well, that's true for any protein. And so that's going to be true for an entire cell. Our cells have a very distinct and unique temperature range, optimal temperature range for growth. Bacterial cells have their own optimal temperature range for their growth, and it has to do with what temperature can our proteins and enzymes tolerate. And depending on what type of bacteria you are, that range can differ. So this is just showing you that for a lot of bacteria, that optimal growth temperature is usually around 37 degrees. That is body temperature. That'll be true for our cells as well. But there are other groups of bacterial cells out there that do better at other temperature ranges. This graph is showing you those temperature ranges for the different groups of bacterial cells. So mesophiles, that range is around 20, maybe a little bit less, up to maybe 45, but optimally, that optimal temperature within that range is about 37. So our cells, us as an organism, we would be known as a mesophile. Bacteria that can infect us would have to be a mesophile as well, because if they need a higher temperature or a lower temperature than our body temperature to survive, they're not going to infect us. They're not going to live in us for very long. Other groups, psychrotolerants, psychrophiles, they like those colder temperatures. Thermophiles like hotter temperatures, and hyperthermophiles like really hot temperatures. Where do you find some of these bacteria in these extreme environments? In the Arctic and the Antarctic for those cold temperature bacteria. And for those that like the hot temperature, there are thermal vents in the ocean floor. There are hot springs. There's hot springs in Arkansas. There's hot springs in Yellowstone National Park. If you've ever seen those, I don't think we have a picture. If you've ever seen those, they're like rainbow colored because of all the different bacteria that can live there, and they're the ones that are providing that color. Other physical requirements, pH, similar to temperature. Extremes in pH will denature proteins and denature enzymes. And so bacteria like a very relatively neutral pH, most bacteria, but not all of them. Our cells like a neutral pH, so we would be called a neutrophile. Acidophiles like acidic habitats, alkalinophiles like more basic along that pH scale. Water. Water is essential for life. We need it for lots of different reactions. When textbooks and such like that talk about osmotic pressure, it really has to do with salt concentrations. So if you put, let's say you put one of your own cells, like a red blood cell, that has a particular salt concentration inside of it that it likes. If you put it in a solution that has a lower salt concentration than what's in the cell, that lower concentration is called a hypotonic concentration. Water will always go to where it's saltier. So if it's saltier inside the cell, water is going to enter the cell and the cell is going to swell. And if you don't have a cell wall, that cell is going to burst. Bacterial cells have a cell wall, so they will just swell. Conversely, if you put that bacterial cell into a saltier solution than what's inside the cell, that saltier solution is now outside the cell. It's hypertonic to what's in the cell. Water is going to leave the cell and go to where it's saltier, and the cell's going to shrink. So it's important for bacterial cells that the environmental salt concentrations are similar to the internal or intracellular salt concentrations, so we don't have this mass movement of water in and out of the cell all the time. All right, so those were some of those different types of growth requirements that you need to consider when you're trying to grow microbes in the lab. What are the oxygen requirements? What are the nitrogen requirements? Temperature and pH requirements? You have or will notice in lab that we have to incubate our cultures at a particular temperature. And the reason for that is so we get optimal growth. So things like that, you need to know all those things if you want to succeed in growing and maintaining those bacterial cells in the lab. Now let's talk a little bit about biofilms. So we said those were collections of bacterial cells. They can be multiple species, and they are kind of feeding off each other in terms of communication, sharing of nutrients, and it ends up being a survival method in a way for these bacterial cells. Some of them do better in numbers, strength in numbers, and they do better in this biofilm than they would on their own. Examples of biofilm in real life. So I mentioned in previous chapters, the plaque on your teeth, that is a biofilm. Soap scum in your shower, that is a biofilm. In the medical community, lots of particularly respiratory devices. If you have a CPAP machine at home, a ventilator in the hospital, if you're on one of something like that, some kind of a breathing device, even like if you're intubated or if you have a feeding tube, that's not even respiratory, a feeding tube, anything like that where the environment is warm and moist, that is a breeding ground for bacteria, and particularly a biofilm, because there is a surface readily available for those cells to attach to and form that film. So how does that actually happen? I'm just going to talk very briefly about quorum sensing. So the way this works is you have these free-swimming bacteria growing. Let's say we're in the mouth. The bacteria are individually around that tooth on their own, but they are subject to stresses on their own. So they end up attaching to a tooth, maybe by chance. And there are multiple different types of bacteria that will do that. When they do that, they start producing an extracellular matrix. So it's almost like these neighboring cells are forming a tissue. You can kind of think of a biofilm as a type of tissue. When they start doing that, they start secreting what's called quorum sensing molecules. Those are signals. They tell the cell to change their biochemistry and their shape to allow them to work together as a team and also to invite more cells to the biofilm. And so more and more cells build up and join that biofilm, and it's almost as if this whole tissue now has taken on its own properties that are different from the individual cells alone. Some microbes will leave the biofilm, some will not. So that's, I think, probably the last time we will talk about biofilms in this class. But that's a very simplistic way of how it works. All right. Okay. So we talked about these growth requirements that bacterial cells need to survive. We talked a little bit about biofilms. Now let's talk about how we grow microbes, particularly bacteria, in the lab. So when we're talking about a culture, we may have covered this in lab already by the time you listen to this lecture. A culture is bacteria growing in some type of medium, whether it's a broth, whether it's solid agar, like in a slant or on a plate. That's a culture. Inoculum is the bacteria part of the culture, and the medium is the food. So when you have sterile medium, like sterile broth, and you introduce bacteria into that broth to create a culture, you are inoculating that medium. That's what inoculation means. And you'll be using an inoculation loop or inoculation needle to do this. Where do you get the bacteria from? So you can get it from the environment. If you're a field biologist and you're trying to study what types of bacteria are in the environment, or if you are working for the water department and you're trying to test the water to see what's in the water, those are some examples of getting that bacteria from the environment. Clinical specimens, if you work in the lab in the hospital and you're trying to figure out what somebody's got, what disease do they have, you need a specimen from them to test. But in the lab sense, we're getting our specimens from our stock that we have stored in our prep area. Big professional microbiology labs will keep stores of bacteria in their freezers that they're studying, and every time they need more, they just go into their freezer and get more. This image, it's a little bit out of place, but when we were talking about colonies of bacteria, these masses of visible cells of bacteria, you can see in this image at the bottom, there are these round red things growing on the plate. Those are colonies. They originally were individual cells, and we incubated them at the right temperature for a day or two to allow those cells to reproduce more and more and more, and now create a mass of cells. So the mass of cells is the colony. you can only see colonies on solid media for the most part. If you're trying to grow bacteria in liquid medium like broth, you're not really going to see individual colonies that way. So if you want single colonies, you need to use agar, solid medium, and you want as much surface area as possible so you can spread those individual cells out. And so therefore a plate is going to be your best bet, the Petri dish. So colonies can have their own morphology. We talked a little bit about morphology in a previous chapter, but we were talking about the shape of individual bacterial cells. So back when we were talking about bacillus and cocci and things like that, those are morphologies, those are shapes of individual cells. But colonies can also have their own morphologies. And it's not just the shape, but also the color of the colony, the opacity of the colony, like how can you see through the colony, can you not see through the colony? Is the colony smooth? Is it rough? How big is the colony? Depending on which species of bacteria you are, the way you grow in a colony is going to be somewhat unique. So when you're trying to identify bacteria, one thing you could look for clues is looking and observing the colonies and looking for the shape. Is it circular? So the ones in this picture I would say are circular. Zoom in on the edge, or the margin we call, the edge of one of these colonies and see, is it actually smooth when you zoom in? We call that entire for the edge. Or is it more wavy? Then we call that undulate. How high is the mound? Is it real high? Is it bumpy? Is it more like a plateau? Is it real flat? Again, size, texture, appearance, pigmentation, things like that. Those are all things you can look for to help you identify bacteria. Okay. All right, so a pure culture. A pure culture is one that only has one species of bacteria in it and a particular strain. When you're working in the lab and you're studying E. coli, you want to make sure that the only bacteria you are looking at is E. coli. You want to maintain pure cultures. One way of doing that is to create cultures from single colonies of E. coli, because you know a single colony is all those cells are all the same. So the cell that leads to a colony is called a colony forming unit, or a CFU. And that colony, the actual colony, is going to be a pure culture because all those cells come from one cell, one parental cell. So being able to isolate single colonies of bacteria ensures that you have pure culture when you're working with what you're working with. How can we do that? There are two ways to do that: the streak plate method and then the pour plate method, although the easiest way to do this is the streak plate method. And so shown here, it's a way of taking a little bit of bacteria from a culture with your loop and successively diluting it across the plate, so that the idea is that you spread out the cells that are on that loop. You want to spread them out as much as possible so this individual cells have enough room to grow into a colony that is isolated from the neighboring colonies. And you can see in this red plate that there are these little dots on the plate. Those are single colonies, and that's what you're trying to do in the lab when you want a pure culture. The pour plate method, you can do the same thing. You often will have to dilute a sample a few times, pour a little bit of those dilutions onto a plate, and use a special spreader instrument to spread the liquid across the plate. And the idea is you're spreading those cells around as well so you can get single colonies. Let's see. All right. What kind of medium is the best to grow those bacteria in the lab? It depends on what you're trying to do. So if you're just trying to grow things, there are some very common media out there that we either know exactly what the ingredients are, or we have a rough idea of what's in there. We just know there's food in there, and almost anything will grow on that. So if you're just trying to grow things, it doesn't have to be necessarily a very specific recipe. You just have to have some general idea of what needs to be in that medium. That a medium that we know exactly what's in it would be called a defined medium. And so there are some out there that you have to follow a recipe, you have to add specific things to it, and there's names for those different media. So in the lab, I know in here they're mentioning nutrient broth. In the lab, we're usually using tryptic soy agar or tryptic soy broth. And I believe that is a defined media. There's a recipe to follow. There's powder you can use to make it. And it's a general kind of multi-purpose media. Lots of things will grow on it. A complex media is something that you don't exactly know the exact chemical composition, but again, it's really there to maintain growth of a lot of different kinds of organisms if you don't know exactly what your particular cells need. Other types of media, selective media. This is useful if you're trying to grow one particular thing and you don't want other things to grow on it. So let's say, you're taking a sample of water and you are very interested in the E. coli that's in that water. All right, but there might be other stuff in that water. So how can you separate them? Well, find a selective media that only allows E. coli to grow on it and nothing else. And anything else that's in that water won't grow. The only thing that will grow will be the E. coli, and then you can isolate it that way. So this is this picture, figure 6.12, examples of selective media. So on the left, this is a medium that's at a pH of 7.3, and you can see a lot of things are growing on it, lots of different sizes of colonies, and other things, you don't know what's in there. There's a whole bunch of stuff. But change the pH of the medium, and now only a few things are growing, in particular some fungi. And so you've selected what you want to grow. You change the conditions of the media to select only what you want to grow. And that's what selective media means. Let's see. Here is an example of differential medium. So this is blood agar. Differential medium, blood agar is one example. There are other examples of differential media, but this is a way, a type of medium that this particular one allows a lot of different things to grow on it, but there's something in the medium that the cells can metabolize, and depending on what species of bacteria you are, you might metabolize whatever's in the plate a little bit differently from other bacteria, and you can visualize those differences. So differential media allows you to tell the difference among different bacteria that grow on the plate. So for blood agar, this is a differential media in that what you're looking for when you use blood agar is you want to know what cells can lyse or break up the blood. There's actually red blood cells in this agar. So some bacteria will break up and lyse red blood cells. Others kind of can do it, and others cannot do it at all. So if you're trying to categorize your bacteria based on this particular characteristic, you use blood agar to figure that out. So there were 3 different bacteria grown on this plate. They all grew. It's not selective. Everything grew, but they all reacted differently to what was in the plate. And there's the differential property of this particular plate. So the organism in the middle here, you can see there's this bright yellow kind of halo and clearing right around where the bacteria is. These bacteria completely used and lysed the red blood cells that were in the agar. We call that beta hemolysis, complete lysis of the red blood cells. The organisms on the left, there's a little bit of a halo, but not a lot. It's more greenish brown than yellow. That was partial breakdown or partial lysis of the red blood cells. So we call that alpha hemolysis. The organism on the right could not use or break down those red blood cells at all. We call that gamma hemolysis. There's no halo or any clearing around that streak. So if you had an unknown bacteria and you streaked it on blood agar, you're going to look for one of these three results, and now you can start classifying your organism, or you learn a little bit more about what your bacteria can do. Some other types of differential media. So this is figure 6.14. It's showing you two tubes. This is a phenol red test. So phenol red is a pH indicator. It is in the broth. Normally the broth is red. There's also some kind of sugar in the broth, some kind of carbohydrate. They don't tell us, at least in this picture, which carbohydrate is in here. I'm just going to pick one. Let's say there's glucose in this broth. You would use this medium if you want to know if your bacteria can ferment glucose or not. Does it use glucose and ferment it as part of its metabolic process or not? This media will tell you that. So the products of fermentation are often acids. So this is the purpose of putting phenol red in the tube, because phenol red is a pH indicator. It will turn from red to yellow in the presence of acid. So the tube on the right is yellow. There was some kind of bacteria added to this tube, allowed to incubate, and the media turned yellow. That means acid was produced because the bacteria that was in this tube fermented the sugar that was in here. Sometimes gas is a product of fermentation as well, and so there's this little tiny inverted tube inside the big tube, and that's called a Durham tube, and it can trap any gas that's produced by the bacteria when they're fermenting the sugar, and you can see that little bubble right here. So in this example, there was probably some bacteria inoculated in the tube on the left, but there was no color change. Everything stayed red, so no acid was produced, no gas was produced, and that means that bacteria did not ferment the glucose that was in this tube. But on the right, this microbe did. So now you can tell the difference between two different microbes based on their use of sugar. Another really useful type of differential medium is called McConkey agar. This is actually, it's kind of not unique, but special because it's selective and it's differential. So this image, this is figure 6.15. This image is showing you just regular nutrient agar on the left. This is like tryptic soy agar. E. coli grows fine, so does Staphylococcus aureus. There's nothing special about that agar. Everything will grow on it. But the image in the middle and on the right are showing you McConkey agar. So E. coli grows on McConkey agar, you can see, and it actually is like dark colored. Staphylococcus aureus does not grow on McConkey agar, and that's because McConkey agar is selective for gram-negative bacteria. Only gram-negatives will grow on McConkey agar. E. coli is gram-negative, Staphylococcus is gram-positive, so it won't grow. So if you have an unknown bacteria and you want to know, is it gram-positive or gram-negative, you could do a gram stain and figure that out. You could also streak it on McConkey agar and see if it grows or not. The problem is, if it doesn't grow, is it because something went wrong, or is it because it really is gram-positive? And so for something like that, it's always best to confirm with the gram stain. The image on the right illustrates the differential property of E. coli. So in this image, E. coli and Salmonella are both streaked on McConkey agar. Both of them grow, so both of them are gram-negative, and they are. However, here's where the differential property comes in. Notice that the two different microbes are two different colors. So E. coli is real dark colored. Salmonella, it looks yellow almost, or colorless really. It's colorless. So there's lactose in these plates in McConkey agar. And the differential property or the differential feature of this agar is basically whether or not your microbe can ferment the lactose in the agar or not. If it can, like E. coli, then the colonies turn that dark color. If it can't, like Salmonella, then the colonies remain colorless. So useful because you can kind of tell what's gram negative and what's gram positive based on whether or not it grows. And if you've got 2 gram negatives on the plate, if one can use the lactose and one can't, they're going to look different on the plate. And it's a useful way of differentiating among 2 different types of bacteria. All right, let's see. Oh, if you are maintaining cultures in a lab long term, is there a way you can kind of preserve them without them dying and without having to constantly maintain and keep inoculating new cultures so things don't run out of food? And you can. You can use refrigeration, you can use deep freezing, you can use lyophilization, which is freeze drying. Notice that in this particular chapter, they're talking about these methods as a way to preserve your microbes. And really what you're doing is slowing down microbe growth. So that is a way of preserving your cultures, but it's also a way of preserving your foods, right? When you are trying to keep your food from going bad, you're trying to slow down microbial growth. So anything that slows down microbial growth is going to make your food last longer. So the same methods for preserving your microbes can also be used for preserving your food, because in both cases, you're just trying to slow down microbial growth. All right, so we're kind of moving on to, we talked about, the things that microbes need for growing, to grow and to live. How can we maintain microbial growth and microbial cultures in the lab? Now we're going to talk a little bit more about like how they actually grow. So we talked a little bit about fission in chapter 11. This is showing you that one cell can turn into lots of cells in a relatively short amount of time. Just a matter of hours, really. So in fission, and then therefore with bacteria, growth is exponential over time. So you get lots of cells from one cell after a short amount of time. What's going on during that time frame or that life of bacterial cells? So this is figure 6.20. This is showing you a growth curve, a population growth curve. Let's use an example of, we took some bacteria on an inoculating loop and we inoculated a sterile broth. Okay, so time is time 0, right when you inoculated your broth. You can measure over time the number of living cells in that broth, and you will see this same kind of shape of the curve. Each phase of the curve could be a different time period depending on the species of bacteria, but you will see this same general curve, and we have names for each of these phases. So when you first inoculate a sterile broth, you don't immediately see an increase in cells yet, because the cells are still kind of getting used to their new environment. They're absorbing their nutrients, starting to metabolize and grow, but they have not yet divided yet. That is called the lag phase, and that's going to be flat because we have not increased any cells. Then cell division starts, and notice how quickly the number of cells increases. This is called the log or the exponential phase. So cells are actively dividing and reproducing here. We know that they're metabolically active too because they are dividing. So if you are, let's say you're trying to make a drug against a particular bacteria, it's during the log phase that bacteria would be most susceptible to that drug. For the most part, depends on what you're targeting. But usually it's when they're actively growing and dividing that a drug will be most effective. Then, so imagine you have a finite amount of medium in that tube, right? A finite amount of broth. Cells are growing and dividing and dividing and dividing, and they're all eating. At some point, they're going to start running out of food. So we get to what's called the stationary phase, which is flat, and we've reached kind of the maximum number of cells that we're going to get. There might still be some cells that are dividing, but at the same rate, there are cells that are now dying because they're running out of food. And then eventually, we're really going to run out of food. There's nothing left, no more cell division, and instead we just see cell death, and that's the decline phase. So again, this is a typical growth curve for a population of bacterial cells. You will see all four of these phases, no matter what bacteria you're talking about, as long as you don't replenish the food supply. You'll see these four phases. The time for each phase depends on the species of bacteria. All right, so can you measure growth? How do we know if our E. coli grew, aside from looking at a plate and seeing, yes, I see colonies, so it must have grown, right? Is there a way, though, that you can get a better sense for how many cells you might have, how much growth? A lot of times when you're looking in the lab, if you've got broth, you're just looking to see if it's cloudy or not. But can you tell between two different cloudy tubes if one tube has more cells than the other? If you're just looking at it, no, you can't. Sometimes we need to know exactly how much is in that tube or how many cells are on that plate. And there are different ways that we can do that. So sometimes we can just estimate. Other times, and I guess let me start by saying, why do we want to know this? So if you're just, taking micro lab, you want to know this because that's the purpose of the lab. But in real life situations, you might want to know, well, how serious is somebody's infection? It depends on how many bacterial cells are in their blood, a little bit or a lot. If you can measure microbial growth, you can tell how well a food preservation technique is working, or how well a disinfectant is working, or an antibiotic is working. You want to know how well those things are working, you have to be able to measure growth, because you want to see, are things growing or not? Or are they growing kind of well with this disinfectant or not at all? That's how we can measure some of these items before we can put them out to market. All right, so how can we do this measuring? So measuring microbes without having to incubate. If you've just got a tube of broth, you can put a drop of that, or I should say culture. Put a drop of that culture on a special counting slide that has grid marks. Look under the microscope and count cells. Sometimes that works. Other times you might have way too many cells, and that's not going to be feasible. So we have other ways of doing that. There are some instruments that can do that direct counting for you. But most of the time, you're going to do some type of counting method that requires you to incubate your microbe first during the process. So there are 3 methods that they're talking about here in the book that require incubation. And again, the purpose is you're measuring growth by counting. You're directly counting something, usually a colony. So the first method is something we do in the lab. This is a serial dilution and a viable plate count. So the purpose of this is you want to estimate or determine how many cells are in 1 milliliter of a culture that you have. You have a tube of E. coli. There are billions of cells in there. You cannot possibly count all of those yourself. You want to know what's just in 1 milliliter of that. And even that is too many to count. So what do? You have to dilute it by 10 multiple times. So we have different dilutions of the same culture. And then we put and spread those dilutions onto individual plates. So we're taking a finite amount. In this example, we've made our dilutions. From each of the dilutions, we've taken 0.1 milliliters of those dilutions and spread them onto individual plates. Spread them out, spread the cells that are in that 0.1 mil out, spread them out on the plate, incubate the plates, and we're looking for single colonies. So in this example, there's the first or second, well actually yeah, the first dilution is only diluted by 10. There's still too many cells to count. Even the 1 in 100 dilution, too many cells to count. But as we get more and more dilute, there are fewer and fewer cells on the plates. And ideally, you want to pick a plate that has a number of colonies that falls in between 30 and 300. If it's more than 300, it's too many to count, and the colonies are going to start squishing together. If it's less than that, it's not really an accurate sample. So in this example, the best plate to choose is the one with 65 colonies. And that dilution was the 1 in 1,000th dilution. So you know how many colonies are on this plate. That tells you how many cells were on this plate. But you want to know how many cells were in your original culture. And this has been diluted. So you have to do a calculation to figure out what was in that original culture, taking into account the dilution that gave you the right number of colonies. So when we do this in the lab, we talk more about how you do that calculation. But again, the overall purpose of this is we're trying to measure how many cells are in this original culture. We have to dilute it first to make it easier to count. Other methods, membrane filtration. So you kind of pour your culture through this kind of paper membrane. The cells get stuck to the membrane. You put this membrane on a plate and incubate them. And if you get individual colonies, you can just count the colonies, and that tells you how many cells were in your original sample. Most probable number. This is a way for estimating how many numbers you have, how many microbes you have in a culture. So you have your undiluted culture, a 1 to 10 dilution, a 1 to 100 dilution. Each of these dilutions gets a 1 milliliter of each of them gets put into 5 different tubes that have a pH indicator, phenol red, which will change color if you have active cells, like we saw before. And then you might see a color change, and the pattern of the color changes. How many yellow tubes do you have versus how many red tubes do you have? That has already been figured out by somebody else. You look at the pattern, and that will tell you how many bacteria are probably in 100 mils of your sample. No calculations necessary. So those were all direct methods of measuring growth. You were counting colonies or counting cells. There are indirect methods too. Turbidity, just looking at, is it cloudy or not? Is the culture cloudy? If it's cloudy, you have growth. But that's all you really can tell. You can't tell more specifics than that. Unless you use a special instrument. So the naked eye probably can't really tell a really cloudy culture versus a less than cloudy culture. But a spectrophotometer can. It can shine light through your culture. The cells in the culture will absorb the light. So the more that the light, more absorbance or more light being absorbed means you have more cells in that plate or in that tube. The fewer cells you have, the less light will be absorbed and the more light will get transmitted through. So this is a way of quantifying exactly how cloudy or how turbid your tubes are. Other indirect methods of measuring growth: metabolic activity. They don't go really into detail, but there's ways to measure how much metabolism is happening in your tube. The only problem with that is your cells have to be alive. And if you've got dead cells in that tube, those will not get counted because they're not doing metabolism. Dry weight. Dry your sample and weigh it. That gives you, it doesn't give you the exact number, but you can get relative numbers if you're going to test more than one sample. Molecular methods, looking at DNA sequences. You can kind of measure how much DNA is in your sample. More DNA means more cells. And I think that is it. So that was a lot of information, but it's all kind of very practical information, all relative to what you'll be doing in lab, what you are doing in lab. What are the best media to use? Well, it depends on what you're working with. What nutrients do I need for my cells? It depends on what you're working with. What's the temperature I need? It depends on what you're working with. And then we talked about all those different ways to actually measure how much growth you're actually getting. So that's it, and I will see you again for Chapter 7.